Hello everyone, hope you guys are doing good. So, we are gonna talk about in this lecture on different laboratory methods that are used in immunology, primarily for like diagnosis or even at looking at antigen antibodies interactions. So, I'm gonna talk focus mainly on tests that involve antigen antibody interaction. So we did have a lecture, I think, when we were talking about the antigens. I touched upon these very briefly. So today we will be talking about in somewhat in greater detail. So for the goal of this lecture is to get the concepts. You don't have to memorize all the details of the tests, but I have put in quite a few pictures in there that will give you an idea where how the tests work. But that's for your knowledge, but not very important for exam. All right, for exam in terms of questions, you should think about there are certain definitions of terms. Those will be there, and of course some examples of different tests. And I will talk about it as we go on. So I do see a typo. Ignore the G's. Okay. Use of antibodies for testing should be the title. So what happens? Why are antibodies or how they are used for testing? Well, antibodies are in general there are different macromolecules or cell surface markers. Those are used to detect different cell types, and these are identified by the terminology CD or cluster determiners. And think of those like CD8 is for cytotoxic T cells, CD4 helper T cells, and in general CD45 is any kind of immune cell marker. Similarly, as we discussed in the previous class about vaccines, how pathogens have their own epitopes. They are used as vaccine. They are used to identify or recognize, and even antibodies have their own receptors and markers. These are in general used for primarily for diagnosed for in, these markers are used by the antigen antibody interaction for to have an immune reaction in the body. However, there are instances when antibodies, instead of just doing regular immune related functions, they are used as labels or markers to identify things. They are sometimes used as traps. They're also used as competitors to look for to specifically reduce the effect of inhibitors. So if there is an inhibitor for a particular antibody, then there is a competing one goes in, if the inhibitor binds to it and the regular antibody is free to do its activity. Antibodies are used for detection of specific cells, looking for localization or movement, and then also quantification, basically how many cells are there, and that will also decide the amount of reaction that has been generated. In terms of historical significance, the first time in 1891 the term anticorpor was used by Paul Ehrlich. He meant the antibody, which is a substance that generates immunity. So the definition that came up was if there are two different substances that give rise to two different anticorpors or antibodies, then those two substances must be different. And the basic idea was to have a lock and key. For every antigen there will be an antibody key that will merge. Later in 1920s, Heidelberger and Avery identified these antibodies as proteins, and Linus Pauling confirmed the lock and key theory. We went over these briefly during our history discussion. Then in 1948, immunoprecipitation was identified as a method for antibody detection, and then in 56, antibodies were identified to be coming from B cell, and then in 76, the gene rearrangements were discovered in antibodies. There are specific characteristics of an antibody. First is specificity. What is specificity? So it's its ability to recognize a particular individual epitope. However, in spite of being highly specific, the antibodies tend to have cross-reactivity. And what do we mean by cross-reactivity? Cross-reactivity is ability of an antibody to bind with to more than one epitope. And that sometimes it happens because there are different epitopes that may be sharing, different antigens that may be sharing their epitopes. Then in terms of binding strength, there there are two terms as well. But affinity, it indicates the strength of binding between the antibody and the epitope. And the other critical term, which is something new for you, we are talking about in this for the first time, is avidity. Avidity is the overall strength of binding of the serum with the antigen. Okay. So let's talk about specificity and cross-reactivity. So even though antibody reactions are highly specific, there are cases where one antibody can react to more than one antigen, and sometimes it's highly completely unrelated one. And when that occurs, it is primarily because those two antigens share very similar, even identical epitope, and the antibody's affinity for the cross-reacting epitope is usually less than the original epitope. So if you see, this is the original antibody, right? So if it's interacting with the specific antigen, which is antigen A, and it's being reacted here. However, it is still possible to have cross-reactivity with antigen B, which has a shared epitope, or sometimes it's not as high affinity, but it can still interact with an antigen C, which has pretty similar, structurally similar epitope, but it might not be identical. So this is the actual specific antigen antibody interaction, and these are instances of cross reactions. One of the examples of cross reactivity is ABO blood group. So for ABO blood group, antigens are glycoproteins that are expressed on red blood cells. There are slight differences in terminal residues of the polysaccharides attached to this surface protein, and those are the ones that distinguish between A and B. And also, for example, a number of bacteria and virus have similar antigenic epitopes, which are a lot of times identical or even have similar looking to the host cell components. So these are the ways they think of the discussion about evasion mechanisms by these virus and bacteria. So they have normal host self-looking antigens that basically have, so the actual antibody may cross react with the host and then let it go, thinking, oh, this is a self, and then the virus or the bacteria, they will survive. And in some cases, the virus or bacteria have been observed to interact and connect directly with the host cell, not the immune cell, but other host cell components. And that sometimes leads to an autoimmune reaction, because while it is the bacteria is binding, it's initially passed on as a self. However, by the time it starts the damage, the actual immune reaction takes place, and it goes to a pretty severe level of autoimmune reactivity. So once again, this is specific antigen antibody reaction, which is really good. However, there are instances where one antigen when the antibody gets confused and joins to a related or similarly looking antigen, and that is not good. That's when cross reactions are not good for the immune reaction as well, immune system. Next term is affinity. What is affinity? Affinity is the strength of the non-covalent interaction between the antigen binding site and one of the epitope of the antigen binding site of the antibody and the epitope. And so low affinity binding between antigen and antibody is, means it's a weak association and it can dissociate very easily. But high affinity binding can remain bound and stays long. So look at this example for look at this for example. Here the antigen antibody are structurally similar and they are fitting really nicely around here. However, looking at this one and the low affinity, structure is not very similar, and in some instances, while they are joining, when they are touching each other, that's basically repulsion and separation. So it's not a strong binding. So affinity is basically the strength of binding. High affinity is well really strong bonding, and low affinity is not that strong. Here in the as in previous equation, k forward and reverse. This k value at equilibrium, if it's around like 10 raised to the power 4, then that's called affinity. It means there is a binding. Stronger affinity, as this k equation keeps increasing, the greater is the affinity moving towards avidity. And if it is at multiple levels, binding in the serum, if there are multiple copies of antibodies binding to the serum with the antigen, is even greater degrees of affinity, of avidity. So F initial just binding is affinity, and then as the binding strength or as the value of the K equation, K at equilibrium increases, it turns from affinity to avidity basically. Antigen antibody binding to moves all the way towards multiple antigen antibody binding, and that binding is in fact binding in serum. So there are different tests that are used to determine or measure the antigen antibody combination. There are primary, secondary, and tertiary tests. Pretty easy to memorize the names. What are the primary binding tests? They are directly measuring the antigen antibody reaction, and these are most sensitive in terms of the amount of antigen antibody. The examples of primary binding tests are ELISAs, radioimmunoassays, Western blotting, fluorescent tests, things like that. What primary antigen or primary binding tests are performed by allowing the antigen antibody without any pressure to combine, and then measuring the complex form. And it usually uses either some kind of dye by tagging them, or enzymes, or even radioisotopes to identify one of the reactants. So you can think of, if you recall the previous equation of antigen plus antibody forming the antigen antibody complex, one of them is tagged, and then the complex is let complex formation takes place of a certain after a certain time, and then unattached ones are washed off, and then at that point the labeled antibody is amount of that is measured to determine how much of antigen has been bound to that. Okay. Then there are secondary binding tests that basically measure the consequences of the antigen antibody interaction. So examples include agglutination. So think of agglutination as multiple antibodies binding to multiple antigens, and then all antibodies binding to each other as well, and they form a complex, and that complex is form a clump basically. That clump is measured And also there are precipitation reactions of soluble antigens. If they are all water soluble antigens, you bring in the antibody, and then the antibody pulls down the soluble material as a precipitate. So, and the supernatant, which is the top liquid part of the reaction, goes out, and the precipitate is measured for quantification. Other examples are neutralization of bacteria, virus, or toxins, and also the complement activation system. These are all secondary binding tests. Examples are agglutination tests, precipitation tests, complement fixation tests. And these are less sensitive to primary binding, but they are much easier to perform. Then there are tertiary binding tests that measure the consequences of immune response inside the body, in vivo. In vitro is in a petri dish in a lab, in vivo is inside the subject or the body. So these are much more complex, but they are much more accurate and practical, they are much more significant actually for in terms of immune response. And these are usually measured for the protective effects of an antibody, how viable that antibody is. In terms of testing methods, as we discussed during the immunoglobulin lecture, there are primary two major types of antibodies: polyclonal, which are isolated from the serum, and they have different idiotypes, and they recognize many different epitopes, and they are less specific and cross-reactive. However, there are monoclonal antibodies which have a single idiotype and highly specific, but they do have lower avidity because they are binding. They do not form complex with each other, and so their binding strength is just at the affinity level, not at the avidity level. Okay. So this is an example. Here is a monoclonal antibody. It is binding to one particular position, while these polyclonal antibodies are binding at multiple places. So monoclonal antibodies are primarily used for diagnostic tests because of their being highly specific to a particular. Polyclonal antibodies, when they are used as reagents, they are used for like purifying the antigen. They're basically used by taking the antigen, cleaning it out, injecting it into a different organism like a rabbit, goat, and then the serum is collected, and then the immunoglobulins or the antibodies are purified. So these are antibodies are their specificity depends on how clean or how pure the antigen is. If the antigen is still mixed, then they won't be as specific. For in monoclonal antibodies, if you recall, are generated using the hybridoma technique primarily, where a mouse is initially immunized, and then its spleen cells are fused with myeloma cells. Myeloma cells are type of cancer cells which have uncontrolled growth, and these clones are then based on the antibody specificity. These are selected, and these are highly specific to an individual epitope. So the polyclonal antibodies are derived from a variety of B cell clones, so they are not as specific, and they are very heterogeneous. They are a mixture of different antibodies, each specific for an epitope. But these monoclonal antibodies are derived from one single B cell clone, and thus they are highly specific for a single epitope. Monoclonal antibodies, you will keep reading those. They are used as diagnostic techniques. They are used for imaging and also for as therapeutic reagents. Just briefly touching upon hybridoma techniques. In general, what is a hybridoma? Hybridoma is a hybrid B cell that is a mixture of an antibody producing B cell and a myeloma cell, which is a cancerous plasma cell. Once those two are fused, that creates a cell type which is called hybridoma, and it has a non unstoppable growth, or it's immortal basically. It keeps growing on and on like a myeloma cell, and it also produces the antibody. So it's basically an antibody producing cell that keeps growing. The clones of these hybridoma cells, which secrete large quantities of monoclonal antibodies, can be cultured indefinitely. Okay. So these clones of hybridoma cells are generating large quantity of monoclonal antibodies. These are different steps, as I mentioned before, just to give you an idea how this things work. This is an image. So think of it, this is an immunized mouse. Its spleen is taken out. The antibody producing lymphocytes are generated, while the myeloma cells that are grown separately are all both cultured in a dish, used fusion, and then they are cloned over and over by selecting just the positive hybrid cells. So at the end of that, these are a whole bunch of positive clones of monoclonal. This is a different visualization of the same process, just to give you a better idea how things are working. So this is again the mouse spleen cell mixed with the myeloma cells, while there are selection of different types of uh, epitopes leading to different clones and different varieties of hybridomas in there, and then each of them are isolated and created into individual hybridomas separately. So all these methods have been developed for using antibodies for diagnostic purposes as well. So and these are used by tests, and these tests in general are called serological tests, which involve the blood from patients, and they are used to identify certain immune deficiencies as well as other diseases. And so what happens is, when there is serological tests performed, it is used to look for whether there is an infection or whether there is any autoimmune diseases or hypersensitivity disease like rheumatoid arthritis and things like that. Or whether to detect if there is an antibody already generated as an impact or as a use of vaccine. And then, like for example, if you don't have your vaccination records, you can get an antibody testing done to prove that you have the antibody already. In some cases, it's used to look for identification of an unknown culture, like what kind of antibody is binding to this particular antigen, which is being cultured in the dish, which is derived from an infection, and depending on the antibody that is binding it to it, the pathogen is identified. There are other situations where the blood type is determined by antibody testing, as well as for other immune deficiencies are also diagnosed when there is a lack of antibodies. And if, irrespective of the infection or the type of antibody put in for testing, if it always comes consistently is coming out as negative, then that person is diagnosed as immune deficient. There are different tests for, like for diagnosis. It is testing for antigen in order to determine the unknown pathogen, and then there is also testing for antibody to determine the pathogen or also to look for deficiency. In terms of research, what happens here is, using these antibodies can identify what are the different cells, their products, or even the cells or macromolecules or the markers are identified as well using these immunological tests or serological tests. Some of the examples for diagnosis, the methods used are hemagglutination, its inhibition, then ELISA, radioimmunoassay, or immunofluorescent assays. Some of the same techniques are also used in research, but there are additional ways that are used, such as cell sorting, immunoprecipitation, immunoblot, and then there are mixed lymphocyte responses and chromium release assays as well. These are for research purposes where the amount and concentration of the antibodies is determined. So let's talk about some of the serological or immunological assays. One of them is based on agglutination. So remember our discussion during I think it was, the humoral immunity lecture, right? So the interaction between the antibody and a particular antigen, when it leads to a clumping, that's called agglutination. The antibodies involved in this are called agglutinins. Antigens involved in this in agglutination are called agglutinogens. Agglutinins are primarily IgG and IgM, which is the first one, and Ig is the largest repertoire of antibody responses. Other tests are precipitation tests, which of course involves precipitation of complexes. So for is used for soluble antigens or sometimes soluble antibodies as well. The once the antigen that's dissolved binds to an antibody, antibody pulls it down, precipitates at the bottom, supernatant is thrown out, and the precipitate is measured for identifying the specific antigen. Then there are colorimetric tests that involve fluorescence or enzyme associated fluorescence. Basically, so due because of reacting with certain enzymes that are present, the molecule becomes fluorescent. These are those are classified as colorimetric tests. Or even sometimes in enzyme interaction can lead to different colors of antibodies, and we will see some examples. These are primarily used in the immunohistochemistry methods. So for precipitation tests, the antigen and antibodies level, the mix that or the complex, it basically will aggregate and it won't stay dissolved anymore, and it will form a visible precipitate. One of the conditions is these antibodies must be bivalent, so basically a single ABS F component won't work. And these antigens must have a multivalent interaction, so basically different immunoglobulins can be used. But most frequently IgM is used because that's the first one that reacts with an antigen. So for example, here are the different ways that antibody complexes can precipitate out at equivalence. So here, if in case of there is excess of antibody, it will multiple it will bind to multiple regions of the antigen. However, if there is excess of antigen, the different regions of the antibodies will bind to each of the antigen. And then if there is a similar number of antigen antibody, which is the equivalence, then the binding is every one. Multiple antigens will be binding to multiple antibodies, but their level overall will remain the same. What is the use of precipitation reaction? Well, first and foremost is the detection, where a precipitate that can be seen by eyes indicates that there is a presence of the particular antigen. And the next step is taking out that precipitate and quantify the amount of antigen present in there by either diluting it or letting it to diffuse after labeling, and look at where it goes around, where it is localized. So this is an example of a precipitin reaction. So for example, this is a free antibodies here, but there is no free antigen because the antigen is bound. There is an excess antibody in this. In the equivalence zone, there is almost equal number of antigen antibody binding, while there are regions, zones where excess antigen is there. And the precipitation, best amount of precipitation is right at the time, right at the equivalence region. In terms of diffusion, there is this method called immunodouble diffusion, where in an agarose gel, a hole is punched, and then the antigen antibodies are placed in the different wells, and then they are led to bind with each other, and then a precipitate is formed when the antigen binds with the antibody and gets deposited in that hole, and then it's the antigen antibody mix, or even if there is no reaction, then just the antigen which will be in there is measured. Then there are the other methods called radial immunodiffusion, where there's an antibody containing gel that gets the antigen in there, and slowly and gradually the antigen concentration around the antibody concentration around the antigen keeps increasing. That leads to the identification of the antigen and also quantification based on the amount of antigen it is absorbing. So if this is the gel that has antibodies spread around, you put in the antigen in here, and gradually the antibodies from all over the start to bind, and you keep gradually increasing the antigen concentration, where there will be a certain point of time when the precipitation starts surrounding the antigen. So with the increased antigen concentration, the ring size of the precipitation keeps increasing. Then there are electrophoresis methods where differentially charged antibody antigen are put in, and based on their diffusion, the binding is measured. The higher, taller the rocket is, or basically taller the band goes due to the electric current, is basically high, that indicates higher concentration of the antigen. So those of you who have taken chemistry, you know what is titration. It's basically keep doing serial dilution of the antigen and then measure the antibody, keep dropping it over at different levels, and wherever it reacts, turns into a precipitate, you identify that concentration as your actual amount. It is not as it's not similar to the actual concentration. It is not the actual concentration, but it's pretty close to that. And the methods that use these are hemagglutination and ELISA. This is a display of hemagglutination. These are the serial dilutions of different levels. So each column here and this well 96 well plate is at a different concentration with positive and negative controls. So, and you keep adding the red blood cell, whether they are binding with the antigen or not. If there is the red blood cell has the antigen, is sensitized to the antigen, it will bind to more and more red blood cells. That will tell you whether they are sensitized or not. So look at this, at sample A is going all the way to 1:32, keeps binding, while sample B is not at all there. Sample C increased at 1:16, and so on. Right? Then there are using immunoassays that use radioisotopes, fluorescent dyes, or enzymes, and they are the enzyme indicator of antigen antibody reaction is called EIA. Then RIA is radioisotope and IFA is fluorescent dyes indicator. So basically, the purpose is you put a label, a tag on this one of the reactants. You can put a tag on either the antigen or the antibody, and then measure the tag for how much of them are bound and how much of them are free floating. Usually, free floating ones are washed away, so the ones that are bound stay have keep showing. The examples are ELISA assay. ELISA, the full form is enzyme linked immunoabsorbent assay. It basically measures highly specific interaction between antigen and antibody in solid phase. It is where the antigen is bound to a surface and the antibody has an enzyme bound to it. And once the antigen antibody react, the enzyme releases a color, and that indicates the presence of the antigen specific to the particular antibody. It is primarily used as a detection. However, sometimes, depending on different modifications of ELISA assay, it can also be used as quantitative by using different titer levels. So this is a demonstration of how ELISA works. So it's like initially the plate is sensitized by the antigen, it's washed and test antibody is added. Then another wash, the ligand is added with the enzyme, and finally the fluorescent is in there. And if there is a reaction between the antibody antigen and the fluorescence is released, then you will see the color. So this is this plate has antigen, right? Antibody binds to it. Now bring in the color coded enzyme in here with the colored ligand, and as soon as if there is a binding, it's not the enzyme is not free floating. When the color changing molecule, the testing molecule or substrate is put in there, this enzyme reacts with that and gives out the color, and that's the indication. So this is the positive control, and these are different concentrations at which the color amount of color determines the extent of reaction. So this is the antigen, antibody bind to it, and the substrate come in. This is the secondary antibody. It has a substrate. When the enzyme is dropped, it reacts and changes color. There are different variations of ELISA, and we looked at these previously, so I'm not going to go into that much detail of these. There are images, so you can look through the steps that take of computed. This is computed ELISA, ELISA spot assays. These are all modifications of ELISA. And then Western blot, we have had, I think I have mentioned previously before, it's basically the fragments, they move on a gel and they migrate based on the size, and then those are measured by a corresponding ladder. And then once these blotting tanks are taken, they are done immunosorting and used for X-ray, and then whatever is the labeled dye that is stuck on that. So if this is the gel, on top of the gel put in a sheet, nitrocellulose sheet, then wash it and add radio labeled the conjugate. So basically that radio labeled conjugate will bind with each of these components or the tags and light up, and that light up will indicate where these different antigens are, and you can easily compare it with a ladder to determine, or a scaling ladder to determine what are the different components. So fragment sizes are in there. So this is a commercial test of called immune card. This is used for testing some, I think it's one of the ways of looking at the different cultures, whether they are test positive or negative, and it is right now a variation of that is used for COVID test. For if you look at the antigen testing in tubes of COVID, You will see there is a control condition and then there is a test condition, test bar. If there is both of them light up in one or other version, then it's positive. If only the control lights up, then it's negative. In some cases, there is no lighting, then it's invalid. It might be due to different reasons, such as not enough reaction material or there is some kind of contamination. Things like that. There are other methods of testing like immunohistochemistry and immunofluorescence. They're basically used in detecting the antigen antibody level within a particular cell or tissue, and these are primarily used for cell surface marker or products that are within the cell, outside the cell, and also other metabolic products that are generated as part of a process of cellular development or metabolism, things like that. What happens is there is an antigen that directly binds to an antibody that is already labeled, and these label the amount of and labeled antibody is measured to identify the antigen's concentration. That's a direct immunofluorescence. However, there are instances where a secondary antibody is added that is labeled, and these secondary antibodies are usually the ones derived from a different species. So it's just not to bring in an additional immune reaction. So the primary antibody is unlabeled, but it binds to a secondary antibody which is labeled. So that's an indirect immunofluorescence. So this is one of the examples here, like how what it is used to identify cell surface markers, right? So for example, the red color here is BCL6, which is a marker for B cells, and the green color here is for IRF4, which is a marker of plasma cells. So it clearly shows that these are here the B cells and these green ones are plasma cells, which are, if you recall, one step ahead, one next step after activated B cells. So there's the immunohistochemistry is used to identify those two localization of these different cells. So this is a more detailed image of second indirect immunohistochemistry where there is a primary antibody, there is a secondary antibody that has a label and a colored product. When there is a substrate goes in, it reacts and generates the colored product. All right. These are other examples like CD45 receptor in B cells are highlighted in brown, while the IRF4 and plasma cells are highlighted here. Those are two different markers of activated B cell and further developed plasma cells. This is an example of enzymatic reaction of caspase-3. Caspase is a mast cell marker. So once it is activated, means these are mast cells are there. And that might as well an indication of hypersensitivity in there. This is a real caspase-3 enzymatic reaction image. There are immunohistochemistry kits that are used to do a quick diagnostic or quick test for presence of antigen, and it basically follows the same procedure. It takes the primary antibody, adds a secondary antibody, adds the enzymatic the tag, the biotin is tagged. So if it's like a biotin related complex is put in, and once the substrate is there, the enzyme reacts. Enzyme tagged biotin reacts with the substrate and color comes out. Yeah. This is a summary of different types of enzyme related assays, what kind of antigen they are targeting, what product they are, or how they are detected, and what are the different variants. This is for your general knowledge purposes. You're not going to be quizzed, you're not going to be asked about that in the exam, but it's possible that you may have to respond to something in the quiz to make sure that you definitely look through the slides. There are other ways where antibodies are used to tag and basically attach to a cell and pull them out. One of the primary most common ways to use that is the fluorescence activated or fluorescence associated cell sorting, or FACS. Then there are other ways like magnetic isolation of the beads, using the magnetic isolation of cells using beads, and also plate related isolation of cells, which is also called panning. This is a schematic image of the FACS sorting where the samples are going in. There's a flow cell is vibrating this fluid that separates each cell, and then there's are lasers that are tagging the cells, and each cell is going through, and based on certain gating strategies, like certain parameters of sizing, they are collected in different tubes, and the liquid is thrown as a waste. So this is, for example, CD3 fluorescence, right? In case of CD and CD3, CD8 fluorescence, using a gating strategy, if you are focusing, you can move these parameters and identify these cells which are CD3, CD8 positive. So basically, these are CD8 T cells. CD3 is a marker for overall all T cells, and the subtype of that has really high CD8 markers are basically your cytotoxic T cells, and you can pull them out. In case of panning, there's a antigen surface antigens of corresponding antibodies are on a plate, and then those are put in there. This mixture of these lymphocytes are applied to the plate to make sure the ones that have the antigen, the specific antigen, are bound, while the rest of them. So here is the antigen with this specific to this particular antibody. So multiple of those, and then there are also antigens which don't have this, which are not specific to this and particular antigen that is on the plate. Those the epitopes are not recognized. So the ones that have these known epitopes, they are bound, while the others are washed away. That's to capture, find the specific antibodies as for quantification, isolation, and quantification of specific type of antigen that is being looked at. This is a process for using magnetic bead separation. So each of the cells that have antigen that have antibodies coated with magnetic beads, and then they are used by a magnet to get attached, while the ones that don't have that particular antigen do not get bound to the magnetic antibodies, and they are free floating. Those are thrown out, and the magnetic bound antigen antibodies are pulled in to take quantify that particular specific antigen. Then there are methods of using the lymphocytes, where things like Li spot uses B and T cell helper functions. These lymphocytes, there are blastogenesis tests for helper T cells, and then there is chromium release assays for cytotoxic T cells. So these are the, for example, the antigen is injected in the mouse from the lymph cells, the primary lymph node cells. Those cells are taken out, they are cultured in the antibody, then some other feeder cells from the mouse are put in the culture, the T cells are activated, and then these are grown and checked for specificity. The lymphocyte stimulation tests involve the whole blood. The centrifuge, the lymphocytes are taken out. They are added in a mix of the culture medium and antigen cell, where they are grown, and then these antigens are taken out, and those are measured for their radioactivity to determine the extent, the amount basically of these antibodies. There's cytotoxicity assays, which is targeting the cytotoxic T cells, where when the cells are put in with the chromium, they are spun and washed. Chromium is taken out. If these are the effector cells, they will die. However, when so those cells that are that apoptosize, They are measured in the supernatant, while the cells, the effector cells basically, will stay bound to the antibody, and they will be in the precipitate. There are complement fixation test is used to look for the heat shock protein or other protein found in blood for presence of complements. And this is where antigen joins with a specific antibody that will create the complement. The will create the complex of complement. There are rapid tests, primarily the antigen-based tests, something like the antigen test of COVID. These are flow-through devices where you just put in a soluble antigen in there in a strip, and they are also sometimes the chromatographic devices. However, there are other methods that take much longer, like Western blot or Southern blot, which you have to let it run for a while, or even the flow cytometry, where you have to look through different to look through the cells that are separated based on their size and the gates, right? Basically by the tags they have been attached to. So to summarize, the antigen antibody interaction is interaction between the enzyme and it that involves a secondary interaction between enzyme and a substrate that helps in either coloring or lighting up. So there are different methods that have been used, that have been developed and used for specific antigen antibody testing, because these are highly specific. However, there are instances where it can cross react, and then there are the groups of immunological techniques that are primary, secondary, and tertiary binding tests that test for the combination of antigen antibody. So, in terms of the exam, what you should focus on, think of the terms specificity, cross reactivity, affinity, and avidity. Then, what are the different methods? Like primary, secondary, and tertiary ones. One or two examples will be good. And then also, what are the different tests and how they work. So not for as much for exam, but for your own knowledge sake. And here are some of the resources. So look through the slides, and the, there will be a quiz. You can answer that by end of Monday, and I will see you all on next Wednesday. All right. Have a good one. Enjoy.